Has recently graduated with a master's degree in biology from Villanova University, where she works with Dr. Samantha Chapman as a member of the Wet Feet Project. Her master's work explores the impact of plant community structure on the encroachment of the black mangroves in the GTM Research Reserve. She is excited to continue working in the Chapman Lab and with the GTM Research Reserve as a project technician on a National Estuarine Research Reserve Science Collaborative Catalyst project called Experimenting with Elevation, building a new collaboration to explore management options for wetland elevation maintenance. And today, Tess is going to share about salt marsh plant community structure and how it influences the success of black mangroves during poleward encroachment. Thank you for joining us, Tess.
Thanks so much, Caitlin. Let's just make sure I can move the screen. There we go. Perfect. So yeah, thanks so much, Caitlin. I'm really happy to be actually able to participate in State of the Reserve this year. Like Dr. Chapman mentioned, sometimes it's kind of hard to get down to Florida and have our schedules line up. And I think the last time that our schedules did line up, I was actually putting this project that I'm talking about today in the ground. So I'm excited to actually be able to talk about it this time. So for the past 2 years or so, I've been working with Dr. Chapman as well as with the Wet Feet Project at GTM. I just finished my master's in December, very exciting. So today I'm just going to kind of walk you through pretty briefly the master's work that I've been working on for the past 2 years, looking at how marsh plant community structure and encroaching mangroves relate to each other in the Florida mangrove marsh ecotone. So I know that a lot of us are pretty familiar with the marshes in GTM and in Florida as a whole, but just as a little recap, this project took place totally in the low marsh of GTM, and the low marsh vegetation community is dominated largely by 2 herbaceous species, which are pictured here. So the first of which is Baetis maritima, which Dr. Dix talked a little bit about, but it's a little succulent species. That's kind of tropical and subtropical, and we're starting to see a little bit more of it throughout the marsh. The other species that's pretty dominant in the low marsh is Spartina alterniflora, the good old salt marsh cordgrass that's pretty ubiquitous up and down the eastern seaboard at least. And these 2 herbaceous species sort of commingle, sometimes in polyculture, sometimes in monoculture in the marshes at GTM. And like Dr. Chapman mentioned, we're seeing more and more of this species up here, which is Avicennia germinans or the black mangrove. We have the lovely Dr. Ed Prophet for scale here, just to show you how large these trees can get. And as an impact of climate change, we're seeing this globally, that plant ranges are shifting because they're not kind of limited by their cold tolerances as much anymore. And such is the case of the black mangrove in Florida. So we're kind of starting to see the black mangrove populate these salt marshes more and more. And in thinking about how these marshes may interact with these mangroves as they continue to encroach, as we face the impacts of climate change moving forward, that kind of got me thinking about how plant community structure may influence mangrove encroachment. Does it facilitate, or maybe there's some competition? So that's kind of the focus of this project. So plant community dynamics are really well studied in places where there's a lot of species diversity, like deciduous forests and grasslands. And the salt marsh is definitely not particularly diverse, right? So community may be a generous term here, because most of the time you have 1 or 2 species in the low marsh. But actually that really low species diversity or species richness can result in more meaningful interactions between species. And in addition to that, this stress gradient hypothesis indicates that increased abiotic stressors can correlate to increased facilitative interactions in plant communities. And salt marshes and mangroves are no stranger to abiotic stressors, right? We've got salt, we've got lots of sun, we've got lots of flooding, all kinds of things. More specifically, Baetis has been, which is pictured up here, Baetis has been implicated as a potential mangrove facilitator, so it may kind of help along mangroves in some way. There's been a couple of studies that have identified it having sort of increased ability to entrap mangrove propagules, so kind of grab them with all of their gnarly little leaves, and then kind of hold on to them, which allows mangroves to establish themselves. Whereas Spartina alterniflora, pictured on the bottom here, it's kind of context dependent. Sometimes it's really good at entrapping mangrove propagules where it's really dense. Where it's really sparse, it may not be. So it's a little bit more geographical and context dependent. But plant community can also impact the below ground environment, of course. And specifically, Batis has been shown to potentially increase available soil nitrogen, which could be utilized as mangroves encroach into salt marsh. And Spartina alterniflora has, there's a cool study that came out last year that indicated that Spartina has soil microbes that are associated with its roots that may inhibit mangrove growth, and specifically can decrease their freeze tolerance, which is particularly interesting right at the kind of, the edge of their range in GTM. And then, of course, like Dr. Chapman talked about, mangroves may also be able to increase soil oxygen. And this is via their parenchyma, which they use as kind of straws to introduce oxygen to their roots. So there's kind of this idea that if mangroves continue to encroach, that'll increase soil oxygen, which will increase decomposition, which may have negative effects on marsh elevation. So I tested 3 hypotheses for this project, and I'll take you through each one pretty briefly. But the first was that mangroves planted in plots with more Batis would have lower mortality rates. So Batis is implicated as this facilitator. Maybe it'll help mangroves survive. The second hypothesis I tested was that mangroves would have lower mortality where there's more plant cover. So the idea that there's this kind of herbaceous canopy that protects mangrove seedlings as they're establishing. And then third, that plots with planted mangroves would have higher decomp rates compared to plots without mangroves. And that's, again, that idea that mangroves may introduce more soil oxygen, which could be reflected in increased decomposition rates. So how did I do it? I established a planting experiment at the middle site of the wet feed project, actually, at which we affectionately refer to as Big Mama because of the big mangrove that's in the middle. But my experimental design consisted of 40 plots, half of which I planted with 10 mangroves each, and the other half remained unplanted for a decomp experiment. And then within those 40 plots, I split those up into 4 plant community treatments. The first was just Batis in a monoculture. The second was Spartina in monoculture. The third was a mixture of those 2 species, roughly 50-50. And the fourth was a mudflat where there was no plant community structure at all. In each of the plants or the plots that were planted, there were 10 mangroves each for a total of 200 mangroves, roughly ages 1 to 3, anywhere from 10 centimeters to 30 centimeters tall. And we planted these in December of, I want to say, 2018, and then let the experiment run till November of 2019, every 3 months coming back and counting which mangrove was still alive, which ones were gone. In addition to that, I also measured percent cover using images and analyzed those in ImageJ. And I also use this really neat method, the teabag index, which uses 2 different types of teabag to get at a decomposition rate. And then it's also an open source data repository, so you can look at decomposition rates from all over the world, which is really cool. Just for some context, for those of you that know these marshes, this is where my plots were in the marsh at GTM. So this is the mangrove affectionately known as Big Mama. You can see some of the wet feet warming chambers in here. And then in the green, we have mixed plots. In the blue, we have Spartina. In the pink, we have mudflat. And way up here in the yellow, we have Batis plots, just to give you an idea of where they're more spatially located. So things happened, a lot of things, some that I expected, some that I definitely didn't expect. So I'll walk you through some of the heavy hitters in terms of outcomes. So I measured mangrove mortality, right? So I counted which mangroves were still alive, which ones were goners. And this figure here is percent mortality over time. So these are the 3 time points that I came back and counted mangroves. Using a repeated measures ANOVA, I found that there was a significant increase in mangrove mortality over time, which you can pretty obviously see on the graph. And then that there was also a difference in mortality across treatments, which was pretty exciting. So Kind of unsurprisingly, we had highest mortality in these mudflat plots. There was no vegetation. Those mangroves did not last long. 100% of them died by the end of the experiment. A little more surprising, 98% of mangroves planted in Spartina plots were also gone by the end of the experiment, which is a little bit surprising considering Spartina is ubiquitous in these marshes, right? Mangroves planted in Beta plots fared a little bit better. They reached about 80% mortality by the end of the experiment. And then lastly, the mixed plots, mangroves planted in mixed plots fared the best. They reach about 68% mortality by the end of the experiment. And the same pattern actually holds for percent cover as well. So across the 3 vegetative plots or treatments, mixed plots had the highest percent cover, and Betis and Spartina were significantly lower. You can kind of see that in these representative images. Spartina looks pretty dense or a little sparse. Betis looks kind of sparse, but the mixed plots are a little bit denser. I didn't take photos of the mudflat plots because I didn't want to bring a camera into a mudflat, and also seemed a little bit like overkill. It was 0% cover, you can trust me. Lastly, we have decomposition. So there was no, similar to Dr. Chapman in the wet feed project, there was no effect of mangrove presence on decomposition, which was pretty interesting. So it doesn't seem like, at least the really young mangroves that were, you know, there and gone in a very short amount of time didn't have an impact on decomp, but differences in plant community structure did have an impact on decomposition. So on the left, we have decomp rate over vegetation. Once again, mixed plots fared the best, mudflat plots fared the worst. Stabilization factor over here refers just to the amount of carbon that was stored during the time when the tea bags were in the ground. There was no difference across these plots, which is particularly interesting considering how different the decomposition rate was. But again, this is just kind of a snapshot of below ground environment. So it's not quite the full story. So just to recap, mangroves planted in mixed plots had the lowest mortality. Mangroves had no effect on percent cover or decomp rate, but mixed plots had the highest percent cover and the highest decomposition rate. And that kind of got me thinking, maybe there's a relationship here. So I plotted percent mortality over percent cover, as well as percent mortality over decomp rate. And I found that as percent cover increases, percent mortality decreases, and the same holds for decomposition rate. So as decomposition rate percent mortality increases, percent mortality of mangroves decreases. And this is an interesting relationship because it seems to me that it could be used as a potential indicator for where mangrove seedlings may have success in terms of planting experiments. But it's also pretty cost effective, right? I took pictures and I planted tea bags, and I kind of got this really interesting gradient in terms of percent cover and decomposition rate. And you can see that these mixed plots kind of fall in this high percent cover, low mortality section of this plot. So what's happening with these mixed plots? That I think is the most interesting part of this, because it's not exactly what I expected.
3 minutes.
Thanks, Caitlin. So increased percent cover, I think, ultimately led to increased protection for mangroves. One thing that I did not measure in this project was elevation, because it wasn't really available to me. But it's important to note that elevation can drive plant community zonation. So maybe where mixed plots ended up being is an artifact of elevation, which I'll go into in just a minute. But also there's differences in below ground environment in terms of decomposition that can also reflect differences in nutrient availability, redox potential, all kinds of things that I couldn't quite get at with this experiment. But I think that there may be sort of a combination of above and below ground effects that may be able to explain this. So if you look at this rather rudimentary cross-section of a marsh, we have the high marsh here with Betis, we have the low marsh here with Spartina, and then there's this overlap. But across the marsh, there's these kind of fluctuations in elevation that, again, may ultimately be driving plant community zonation. So where we have an overlap with Betis and Spartina, it could just be that that's where the the hydro period is not quite as long. And ultimately, that may be driving mangrove establishment. It's kind of, maybe it's the sweet spot for mangroves to establish. In terms of below ground effects, Betis has a little bit more of a taproot style root structure, whereas Spartina has a very fibrous root mat. And as Betis moves into these areas that are a little higher in elevation that suits them, they could be kind of breaking up that Spartina root mass, which provides a little bit more space to allow for mangroves to establish themselves. So we kind of get this above and below ground combination effect. So just to wrap up, it seems like mangrove seedling mortality correlated with both percent cover and then decomposition rate. Mixed marsh treatments can increase mangrove survivorship. And like I said, that may likely be due to a combination of both above and below ground effects, but just due to the things that I measured in this project, that whole story may not be there just yet. And lastly, this idea that establishing foundation marsh species may be useful in restoration context, which there's a paper by Yando in 2019 that expands on this idea, but it's this concept that if the marsh is healthy, then either mangrove encroachment may occur at a more readily, or you can use this as a means of preparing somewhere that mangroves have died back. So effectively, if you build it, they will come. And with that, I'm happy to take any questions, but I'd love to thank all the folks at GTM who helped make this project happen, as well as everybody at Villanova. And I'm really excited to keep working with GTM through this experimenting with elevation project. So yeah, and I'll also pop my email in the chat box because I had to kind of move through things a little bit more, but if you have any questions, I'm happy to answer them.
Thank you so much, Tess. I love the photo in the marsh covered in mud. For those of you who haven't had a chance to experience that, make sure you try to volunteer at the GTM Research Reserve. We'll get you covered in mud. We do have a couple of questions in the chat box, Tess. How did you separate out mortality due to transplant?
That's a really good question. I didn't specifically separate out mortality that was simply due to transplant, just because of timing, because we were going down in 3-month increments, and this project was only running for less than 12 months. I think it ran for about 11 months. So it wasn't exactly clear where mortality was due to transplant. So that could certainly be a factor that's kind of sort of built into those mortality measurements. That's a good point.
Great question. Thank you. Do you mind or could you expand a little bit more on how the decomposition rate is measured with the T-bag index?
Absolutely. It's a really cool means of measuring decomposition rate because you have 2 different types of tea, a rooibos tea and a green tea, which have different fractions of labile carbon. So you put these 2 tea bags in the same plot, and then you leave them there for about 90 days. You take them back out and you wash them and weigh them, etc. And since those 2 tea bags have different fractions of labile carbon, they decompose at different rates. So using those 2 measurements, you can calculate the decomposition rate using just one time point rather than over a longer period of time like other tea bag methods use. There's a really great, I'll put it in the chat box, but there's a really great resource for this. It's called teatimeforscience.org. They've got all the information on it, and they even help you calculate out your decomposition rate. So it's a really cool method.
Thank you so much for sharing that. You can have tea in the office and then go out in the field and have some tea as well.
Exactly.
All right, we do have time for another question. If anybody is typing into the chat box, or if you would like to raise your virtual hand. And it looks like, Tess, that Nicole added the teatimeforscience.org link in the chat box for you.
That's fantastic. Nicole is also using that method for her project as well. So thank you so much, Nicole.
All right, Tess, thank you so much for sharing more about your project with us this morning.